Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel A-Level Biology, and the first topic, which is topic 1A, we're going to be looking at the structure and properties of water. We're going to be looking at carbohydrates, lipids, as well as proteins. Let's begin with the first part. So here we're going to begin by looking at the atom. Of course, an atom is the basic unit of all elements, and we know if two or more different atoms react together, a compound is going to be formed. You can see this is a compound. When two or more atoms that are different react together, we get a compound. Also, we know that inside this atom, there is a nucleus. And if you see, if this is an atom, there is going to be a nucleus inside. Around the atom, there are going to be some shells surrounding the nucleus. And in these shells, we're going to have some electrons. Inside the nucleus, we're going to have protons that are positively charged. And we're going to have neutrons that have no charge. The neutrons, we also have to know these electrons are going to be negatively charged. So that is what is explained here. You need to also know that an atom with a full outer shell of electrons is stable and does not react. However, most of these atoms do not have full outer shells, so they end up participating in reactions. In ionic bonding, the atoms involved in this reaction give or receive electrons. One atom gains one or more electrons and becomes an anion, which we call the negative ion. And then the other atom loses one or more electrons and becomes a cation, which is the positive ion. So the forces of attraction that form between the cation and the anion are what we call the ionic bond. So to demonstrate this, we can see we have atom A, which has one electron in the outer shell, and we have atom B, which has 7 electrons in the outer shell. When this electron is lost, this becomes a positive ion, the cation. Of course, losing this electron, I eliminated the outer shell that will no longer have electrons. So you see we do not have an outer shell here. I also put square brackets to show that this is going to be an ion. I put a positive charge because it's become a cation. The other has gained 1 electron. From here, you can see the color is there. And you can see it has 8 electrons in the outer shell. And the charge is displayed here outside the square brackets. The bonding between the two is what we call an ionic bond. In covalent bonding, the atoms involved in the reaction share electrons, and then the covalent bond is very strong, and the molecules formed are usually neutral, meaning they are not charged. Also, in some covalent compounds, the molecules are slightly polarized, and so they could have partial charges. An example I can use is HCl. Because electrons in this bond are closer or drawn closer to chlorine, this gains a negative charge, which is partial, and this gains a partially positive charge. So this tendency to have partial positive and partial negative is what we call polarity, or we can say there are dipoles that are formed. So some covalent compounds have dipoles in them, and some do not. In atom A, we have 7 electrons, and atom B, we have 7 electrons. When these share one pair of electrons, which is shared, you can see here, we can see each has 8 electrons in the outer shell, and this force of attraction between them is what we call a covalent bond. Next, we're going to look at important inorganic ions in biology. I'll begin with the anions. Anions are those that are negatively charged after they gain electrons. The first one is a nitrate, NO3-, or you could write it as NO3, the negative is like that. So these nitrate ions are used in the formation of DNA. You know DNA has nitrogen bases, and in those, the nitrogen comes from a nitrate. Also, amino acids require a nitrogen atom, and that should come from a nitrate. Proteins also require a nitrogen atom. Actually, anything that comes from proteins should always have a nitrogen. You go with hormones, enzymes, and so on. Those are all going to be made in the presence of a nitrate ion, or at least in the process of making them, a nitrate ion could have been used. Then when we go to phosphate ion, you guys know it's going to be used in ATP formation of ADP. This is DNA, formation of DNA, as well as RNA, and so many more. All of them have a phosphate group. Then chloride ions are used. Of course, sweat contains chloride. Transmission of nerve impulses requires chloride ions, and some other secretions also require chloride ions. The hydrogen carbonate ion is important in the buffering of blood to prevent it from becoming too acidic. When we go down here to the cations, which are positively charged, important cations, we have sodium ions. These are used in the transmission of nerve impulses. They're useful in sweating. You know, sweat contains sodium chloride. They are also used in many secretory systems. Calcium ions, we write them as Ca2+. These are used in muscle contraction, they're used in bone formation, they are useful in calcium peptide formation, and you know calcium peptide is found within the middle lamellae in plant cell walls. We also have hydrogen ion. This is important in respiration. This is also important in photosynthesis as well as in pH balance. Then we go to magnesium ions. These are used in making chlorophyll, which is useful in plants during photosynthesis. The next part, we're looking at the chemistry of water as a whole, and here we're going to be interested in the polarity of water as well as the hydrogen bond that forms between two water molecules. Hydrogen bond. So here I began off by saying water is a simple molecule. Of course, two hydrogen atoms and one oxygen atom interact together in covalent bonds to produce a water molecule. Water is polar because the oxygen atom in each water molecule is partially negative and the hydrogens are partially positive. So this creates an attraction between the partially negative oxygen of one water molecule and the partially positive hydrogen of another water molecule. And that attraction is what we call a hydrogen bond. We also need to know that this hydrogen bond is not as strong as ionic bonds as well as covalent bonds, but it creates some interaction which is useful in determining the properties of water. Looking at the importance of water, here we have the property of water and its importance. So these two could be combined. We can say water is a polar molecule and also water contains dipoles. This is useful because it is polar, so it will dissolve many ionic substances. For example, you have sodium chloride, which can be dissolved. We'll have sodium ions as well as chloride ions. And also water contains dipoles. These make it an excellent transport medium because it's going to dissolve many substances. The other property is, as water cools to 4 degrees Celsius, it reaches its maximum density. Thus, ice has a lower density than water. This is the reason as to why ice floats on the surface of water in cold days, and this prevents water underneath from freezing, allowing survival of marine life. To explain further on that, we can see when winter comes, lakes and rivers can be frozen like that, but down here, the water is still okay and actually warmer, so fish and other aquatic life can still survive even if the water has frozen on the surface. This property allows the frozen water, which is actually colder, turning into ice to be on the surface, preventing the other part from cooling down, so that is advantageous for maintaining aquatic life. The next property is it has a higher specific capacity, and this ensures that it is slow to absorb as well as release heat. The next property, it cannot be compressed since it is a liquid. This is useful in many hydraulic mechanisms in living organisms. The next property is water molecules are cohesive. Cohesive means they can stick to each other, meaning one water molecule interacts with the next water molecule. So this is very important in the transport of water from the roots through to the leaves. The next property is water molecules are adhesive, meaning these molecules attract to other substances or other molecules. This is important in plant transport systems because it allows the water molecules to go through the xylem in the transpiration stream. And also, water has a very high surface tension. Surface tension can be expressed when the attraction between water molecules is greater than the attraction between water molecules and air. This is important in plant transport systems, and it also affects life at the surface of ponds, lakes, and other water bodies. Next, we're looking at carbohydrates. Carbohydrates are useful in cells as a source of energy, and they form an important part of cell walls of plants, fungi, as well as bacteria. The basic unit of all carbohydrates, they consist of carbon, hydrogen, as well as oxygen. There are 3 main groups or classifications of carbohydrates. They can be categorized into monosaccharides, disaccharides, as well as polysaccharides. Monosaccharides are made up of 1 sugar molecule. Disaccharides are made up of a combination of 2 sugar molecules, and polysaccharides may contain many. Beginning with monosaccharides, they are simple sugars with a general formula. I'm going to write it as CH2O and then N. So these include triose sugars. This is where N is equal to 3. So an example is going to be carbon 3, hydrogen 6, or 3. That is an example of a triose sugar. You are not required to know the details of this at your AS level. Then we go to the pentose sugars. These include ribose as well as deoxyribose. This is found in RNA and the other is found in DNA. So you need to know the basic structures. I put here an example which is ribose. You can see it's like that. Also, I included hexose sugars where N is equal to 6. An example is glucose where we have carbon 6, hydrogen 12 or 6. This is when I substitute n is equal to 6, I get something like that. And this could be glucose, galactose, or fructose. So you need to remember the structure of glucose. And again, we have two different forms of glucose. We have alpha glucose and we have beta glucose. Usually in Unit 1, we're more interested in alpha glucose. And then in Unit 2, we focus more on beta glucose as we talk about cellulose. So please remember these basic three structures: ribose, alpha glucose, as well as beta glucose. And you should know the difference between alpha and beta is just, you can see here we have the hydrogen up and the OH down. Here we have OH up and hydrogen down. So memorize one and then just switch to the other position in order to not forget. Moving on to disaccharides. When two monosaccharides join in a condensation reaction, meaning loss of water, a disaccharide is going to be formed and a water molecule is going to be released. The link formed between the two monosaccharides results in a covalent bond known as a glycosidic bond. We'll talk about that a little bit later. Benedict's solution is used to test for reducing sugars, and as you know, all monosaccharides and some disaccharides are reducing sugars. Sucrose is not a reducing sugar. So to test for these reducing sugars, we use Benedict's solution, and if a precipitate is formed, a color change from blue to orange could be a positive result. But remember, the color could be red, it could be orange, it could be yellow, it could be green. So any of these colors, if they are formed, that could be a confirmation of a reducing sugar because these colors represent different concentrations of reducing sugars. Blue means no reducing sugar, green means there is kind of a lower concentration, yellow, increase in concentration, orange, and then red will show the highest concentration of reducing sugar. So down here is a table that shows the name of the disaccharide as well as the component monosaccharides that are contained within that disaccharide. In maltose, we have two alpha glucose molecules, and then sucrose is made from one alpha glucose and fructose, while lactose is made from an alpha glucose and galactose. When we go to polysaccharides, polysaccharides are made when many monosaccharide units join together again in condensation reactions to create glycosidic bonds. So some of these polysaccharides include, we have starch, we have cellulose, and then glycogen. So we will talk more about cellulose a little bit later, but we're going to be interested in starch as well as glycogen. So I'll begin with starch. Starch is a mixture of two compounds. These include amylose as well as amylopectin. Amylose is an unbranched polymer of glucose units. We know amylose contains only 1,4-glycosidic bonds, as we can see here. The 1,4-glycosidic bond is formed between a carbon 1 and a carbon 4 on different glucose molecules. Now, as these glycosidic bonds form in amylose, a long unbranched chain is going to result, and this is going to spiral as it lengthens. You can see this is a spiral formed as the lengthening occurs because only 1,4-glycosidic bonds are present. In amylopectin, a branched polymer of glucose units is going to be formed. It contains many 1,4-glycosidic bonds and a few or some 1,6-glycosidic bonds. So this results in something that is going to be branched, as we can see here. In amylopectin, we're going to see these 1,4, 1,4, 1,4, and then we have some 1,6-glycosidic bonds that occur occasionally, creating a structure that is going to be branched. So this is a summary, and this is extracted from your textbook. We have amylose, which is going to look unbranched. It can spiral as it lengthens. We have amylopectin, which is going to be branched in this manner. So here I also included glycogen. Glycogen has a structure that is similar to amylopectin, but we have more branches. You can see it's further branched in comparison to amylopectin, so it has more 1,6-glycosidic bonds, although it also has 1,4-glycosidic bonds. Glycogen is also made up of only alpha glucose molecules. After carbohydrates, we go to lipids. Lipids contain many carbon-hydrogen bonds and almost contain no oxygen. Lipids make part of the cell membrane. We'll talk about this later on in the next topic. Talking about the phospholipid bilayer, you'll see lipids make up the cell membrane. Lipids, especially triglycerides, store 3 times as much energy as that stored in carbohydrates. So lipids are better for energy production. The main groups of lipids include fats and oils. We know fats usually come from animals and oils come from plants. Also, fats are solid at room temperature, while oils are liquids at room temperature. They contain 2 organic chemical substances, which include fatty acids as well as glycerol. This is kind of an alcohol. These are combined by an ester bond. So a glycerol has the chemical formula, which is this. The structure of a glycerol is like CH2OH. We have a CHOH, and then we have a CH2OH. So I put a 2 there. So that gives us a molecular formula, which is that. Fatty acids have a long hydrocarbon chain, and then they have a carboxylic group. You could say it's going to be RCOOH at the end, but this could be long or it could be shorter, but usually it's a long hydrocarbon chain. These fatty acids can vary in two ways. It could be the length of the carbon chain, which is going to be that. It could be a shorter length or a longer length. And then some of these groups here, the R groups, could be saturated or unsaturated. If it's saturated, it means it contains carbon-carbon single bonds only. And if it's unsaturated, then it contains carbon-carbon double bonds as well. So here is a structure of a triglyceride. I'm going to show you how it is formed from a glycerol and 3 fatty acids. In each triglyceride formed, we're going to have 3 condensation reactions occurring. We can see this is one fatty acid, the next fatty acid, and the last fatty acid, where we have one glycerol molecule. So between the carboxylic part and the alcohol part loses a hydrogen, while this loses OH to form water. The same occurs here and here. And those 3 are going to be linked up in what we call an ester bond, and a triglyceride is going to be formed as we see here. Now you need to remember that this condensation reaction, as well as the hydrolysis reaction, meaning when you add water, are going to be enzyme controlled. So when they ask you to describe how a triglyceride is formed, you have to say it's going to be formed between one glycerol and 3 fatty acids in a condensation reaction that is going to be enzyme controlled. And then 3 ester bonds are going to be formed in that triglyceride. Next we go to proteins. We need to know that all proteins contain carbon, hydrogen, oxygen, nitrogen, but many proteins also contain sulfur. Proteins are made up of many monomer units called amino acids, and these are going to be joined together in condensation reactions. And again, you need to know these are going to be enzyme controlled. And when this occurs, a peptide bond is going to be formed. So if you connect 2 amino acid molecules, you form a peptide bond between them, and the whole thing is going to be called a dipeptide. Naturally, there are 20 different amino acids, and these can be used to make enzymes, hormones. They can be used to make hair, nails, antibodies, hemoglobin, and so on. If you're asked to test for presence of a protein, you will need to use Buret's reagent. You do not have to talk about the details of whatever you mix, but if you want, you could say 5% weight by volume of sodium hydroxide and 1% weight by volume of copper 2 sulfate. However, you could just say Buret's reagent. The result is going to be that a purple color is going to form, and that will indicate presence of a protein. Since we said proteins are made up of amino acids, we need to understand what amino acids are. Amino acids have a structure where we have an amino group and a carboxylic group attached to a specific carbon atom. We have the R group, and these R groups are going to vary. So meaning this part, that part, and that part are the same in all amino acids. The variation is going to be in the side chain, talking about naturally occurring amino acids. So a group known as the R group varies between amino acids. Some amino acids contain sulfur and selenium in their R group. A peptide bond is formed between 2 amino acids as they join, and the molecule formed is going to be a dipeptide. So meaning one amino acid and the next amino acid form a peptide bond after the condensation reaction occurs, and this whole unit formed is going to be called a dipeptide. After water is lost in the condensation reaction, if many of these are going to be connected together, then we can form a polypeptide. This polypeptide then forms a protein when the structure of the chain changes by folding or coiling or associates with other polypeptide chains. We can see this in collagen. Other bonds in the protein structure include hydrogen bonds, disulfide bonds, as well as some ionic bonds, although we can also talk about London forces or what we call dipole interactions. Here's an example of how a dipeptide can be formed. And again, one amino acid and the next amino acid. Again, this is extracted from your textbook. So from one amino acid, we have a carboxylic group, and that carboxyl group has OH. So the OH of one carboxylic group is going to interact with a hydrogen from the amino group, and water is going to be lost. Then this nitrogen is going to connect to that carbon, forming what we see here. So that bond is going to be called the peptide bond, and this whole structure is what we call the dipeptide. This is a reversible reaction. We can see if water is added under right conditions, then this bond is going to be broken so that we form the individual amino acids. And then if water is lost through a condensation reaction, we're going to form the dipeptide. These two reversible reactions are going to be enzyme controlled. So here I'm going to introduce the bonding in proteins. I'm going to be talking about ionic bonding, covalent bonding, as well as hydrogen bonding. Remember that proteins contain the hydrogen bonds, a disulfide bond, ionic bonds in their 3D structure. These formed bonds depend on the R group within the amino acid that the protein contains. It means if there is no sulfur, then there is no reason for forming the disulfide bond. Hydrogen bonds will form between differently charged hydrogen and oxygen atoms in a protein. We saw this in the beginning of this video. You can refer back if you want to understand how hydrogen bonds form. Disulfide bonds form between two cysteine molecules that are close together. So disulfide bonds are much stronger than hydrogen bonds, but they happen less frequently. Also, ionic bonds can form between some strongly positive and strongly negative amino acid side chains. Going to the structure of proteins, we're going to talk about the primary, secondary, tertiary, as well as quaternary structure. The primary structure contains a sequence of amino acids arranged to create a polypeptide, as you can see here. Again, this is extracted from your textbook. The secondary structure contains repeating pattern of the polypeptide structure. The most common types of secondary structures are going to be the alpha helix and then the beta pleated sheets. Here, hydrogen bonds form between the carboxyl and the amino groups of different amino acids within the backbone. This means not within the R groups. There is no interaction between the R groups when you're talking about the secondary structure. I emphasize it here, you have to know. The secondary structure does not involve interaction from the R groups. So we can see here we have the alpha helices as well as the beta pleated sheets. In the tertiary structure, interactions between R groups of different amino acids lead to folding of the secondary structure as hydrogen bonds, ionic bonds, disulfide bonds, and dipole interactions can form. We have R groups interacting so that the structure, which is the secondary structure, can fold further. In the tertiary structure, hydrophobic R groups interact with each other and hydrophilic groups can be exposed because the situation or the environment in which the folding occurs has water in the surroundings, so usually hydrophobic groups are going to fold inside and hydrophilics are going to be exposed on the outside. Next is a quaternary structure. This is a 3D arrangement of more than one polypeptide chain with hydrogen or dipole interactions. Here we can see we have 3 tertiary structures interacting together to produce this quaternary structure. And in this situation, we're going to have hydrogen bonds forming, dipole interactions forming, and so on. I'm going to talk about fibrous proteins as well as globular proteins. The reason for this is because there are many questions asking about collagen and describing the structure of collagen. So this is going to be my emphasis here. We have to know that they have little or no tertiary structure because, of course, they're going to be arrangements in fiber-like form. So they are long parallel with occasional cross-linkages to form fibers. As we can see here, these are fibers. They are insoluble in water and very tough. They are found in hair, nails, feathers, horns, bones, and so on. The primary structure of collagen consists of repeating sequences of glycine with 2 more amino acids, which are usually proline as well as hydroxyproline. Again, this question is very common. You need to know glycine, proline, and hydroxyproline. These are going to be held in a triple helix like you can see here. So I assume this is going to be the primary structure containing many repeating sequences of the 3 amino acids. And then creating a polypeptide, and again 3 polypeptides are going to come creating a triple helix held together by hydrogen bonds, and these will form collagen molecules that are assembled to form collagen fibers that then are going to be arranged to form a collagen fiber, as you can see here. The key thing is, remember, the primary structure is going to be a sequence of 3 amino acids, usually glycine and 2 like proline and hydroxyproline, that are going to be repeating and repeating and repeating. Then 3 of those can be arranged in a triple helix, as we can see here, to create this structure, and that is going to be used to make the collagen molecule that can be assembled to form the collagen fibril and then the collagen fiber. Then we go to globular proteins. The example is hemoglobin. These are complex tertiary and sometimes quaternary structures. They will fold into spherical shapes like we can see here, and some R groups are going to be hydrophobic while others are going to be hydrophilic. Their hydrophilic R groups interact with water, but these proteins are never soluble due to their big structure. They just form colloids. You also need to know that globular proteins function as enzymes, antibodies, and some do function as hormones. A typical example is hemoglobin, and this is what we see here. These are the 4 polypeptide chains, and then we have 4 heme groups that contain iron. Lastly, we go to conjugate proteins. These are proteins that are combined with other prosthetic groups, and this is going to enhance their role in performing specific functions. For example, glycoproteins. You can see the name glyco and protein. Glyco comes from a carbohydrate, so a glycoprotein has a carbohydrate prosthetic group. Examples, we have mucus and then synovial fluid. We also have lipoproteins, and again, this is the lipid part and proteins, so these are going to be formed when proteins are conjugated with lipids. You also need to know that these lipoproteins help in transportation of cholesterol in the blood. In this transportation process, the lipid part of a lipoprotein is going to combine with the lipid part of the cholesterol and then enable its transportation. Here we will talk about LDL and HDL, but I'll transfer these to cardiovascular disease so that we can go into that in detail. So this brings us to the end of topic 1A. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.